Hi everyone, today I'm going to explain about toxicity testing on sex-linked recessive lethal test. Before moving on further, what is lethal mutation? Lethal mutation is a change in the genome which, when expressed, causes death to the carrier. Recessive mutation is a change in the genome which is expressed in the homozygous or hemizygous condition. Sex-linked genes are present on the sex X or Y chromosome. Sex-linked genes in the context of this guideline refer only to those located on the X chromosome. Mutagenicity assays is to identify and analyze agent with genotoxicity. The type of test that used to determine the toxicity is in vitro test by using Drosophila melanogaster. The purpose of using Drosophila melanogaster is because it can detect the occurrence of mutation at both point mutation and small deletion in the germline of the insect. This test is a forward mutation assay capable of screening for mutation at about 100 loci on the X chromosome. This represents about 80% of all chromosome loci. The X chromosome represents approximately 1/5 of the entire haploid genome. The principle of this testing is that mutations in the X chromosome of Drosophila melanogaster are phenotypically expressed in male carrying the mutant gene. When the mutation is lethal in the hemizygous condition, its presence is inferred from the absence of one class of male offspring out of the two that are normally produced by heterozygous female. The SRRL test takes advantage of these facts by means of specially marked and arranged chromosomes.
Okay, so let's proceed with the mechanisms of this test. Wild-type males are treated and mated to appropriate females. Female offsprings are mated individually to their brothers, and in the next generation, the progeny from each separate dose are scored for phenotypically wild-type males. Absence of these males indicates that a sex-linked recessive lethal mutation has occurred in a germ cell of the P1 male. Next, for the Drosophila stocks, the Muller 5 stock may be used. Other appropriately marked female stocks with multiple inverted X chromosome can also be used. Now, let's proceed how this test chemical is being carried out. First, the test chemical should be dissolved in water. Compounds which are insoluble in water may be dissolved or suspended in appropriate vehicles such as mixture of ethanol and 2,60 or Hg, and then it will be diluted in water or saline prior to administration. For the initial assessment of mutagenicity, it is sufficient to test a single dose of the test substance for screening purpose. The dose should be the maximum tolerated dose or that produces some indication of toxicity, or should be the highest dose attainable. For dose response purpose, at least 3 additional dose levels should be used. Next is for the route of administration. We can use orals or by injection or by exposure to gases or vapors. Feeding of the test compounds may be done in sugar solution. When necessary, substances may be dissolved in 0.7% sodium chloride solution and injected into the thorax or their abdomen. Okay, next is control group should be carried out in this test, such as concurrent controls, positive controls, or other positive controls and negative controls. Okay, now let's proceed to the procedure of this test to the Drosophila melanogaster. First is wild-type males, which is 3 to 5 days old, are treated with the test substance and mated immediately to an excess of virgin females from the Muller 5 stock or from another appropriate mark with multiple inverted X chromosome stock. Then, the females are replaced with fresh virgins every 2 to 3 days. The offspring of these females are scored for lethal effects, corresponding to the effects on matured sperm, mid or late-stage spermatids, early spermatids, spermatocytes, and spermatogonia at the time of the treatment. Heterozygous F1 females are allowed to mated individually with their brothers. In the F2 generation, each culture is scored for the absence of wild-type males. If a culture appears to have arisen from an F1 female carrying a little in the parental X chromosome, such as no males with the treated chromosomes are observed, daughters of that female with the same genotype should be tested to ascertain whether the lethality is repeated in the next generation.
Now we will have a look at how this test result is interpreted. The criteria needed for a positive result is a statistically significant dose-related increase in the number of sex-linked recessive lethals. Next, the detection of a reproducible and statistically significant positive response for at least one of the test points. Moving on with the non-mutagenic result. A test substance do not produce either a statistically significant dose-related increase in the number of sex-linked recessive lethals or does not produce a reproducible positive response at any one of the test points. Now let's have a look at the advantages of sex-linked lethal test. Firstly, it is an inexpensive test. Next, the sex-linked lethal test is easy to be performed. Third, the use of fruit fly Drosophila melanogaster as the model organism brings about a few advantages such as it has a short life cycle, ease of culture and maintenance, it has a low number of chromosomes, and it has a small genome size. There are a few limitations of sex-linked lethal tests. Firstly, it takes time to obtain the final result. This is because the fruit fly Drosophila melanogaster needs to undergo a complete life cycle. Besides that, the use of Drosophila melanogaster has a few disadvantages such as first, Drosophila melanogaster only has 4 pairs of chromosomes. Second, anatomy of flies and humans are very different.
So I think that's all from our group. Thank you.